So I'm Peter Francis. I'm the Chief Scientific and Chief Medical Officer at Therapeutics, which is a Bay Area-based new biotechnology company working in the optogenetic space for vision restoration. And at Ray Therapeutics, our vision is to restore patients' vision with retinal degenerations. We have a lead asset, which we're calling RTX-015, which is an AAV-based gene therapeutic, which we believe will be the first to market and actually the first in class precision optogenetic medicine in the optogenetics field. Part of the reason why we're going to be first to market is that we have a very de-risked development program, and I'll show you some of the preclinical science here. We hope to be in clinic by this time next year. Another key to our success is our management team, who are all world-leading regenerative medicine specialists who have done this for many decades in terms of translating these therapies forwards. We're post-seed round. Our lead investor was 4Bio Capital, and we recently received a California Institute of Regenerative Medicine Award, a Tran-1 grant with a top score for our particular program. So very grateful to CIRM for that. So why should we be excited about optogenetics? You can see on the schematic here, which we've adapted from the published literature, how the retinal degenerations like retinitis pigmentosa are unified by the loss of photoreceptors, rods, and then cones, and then the rest of the retina, with the exception of the retinal ganglion cells largely, wither and degenerate. Most of the other approaches, which are pretty exciting, gene replacement, gene editing, neuroprotection, cell therapy, are really going to benefit those patients who still have some existing surviving photoreceptors. Since they don't regenerate, these approaches are mainly aimed at slowing down degeneration or arresting degeneration. So hugely valuable to patients, but there's a large proportion of the patients with RP, for example, maybe 15% or so, who have no really surviving functional photoreceptors. And this is really where optogenetics fits in its nice niche. And hopefully we have the next generation approach that will realize the full potential of optogenetic therapy for vision loss by bringing back vision to those patients who have severe blindness from these conditions. So indeed, our lead indication, RTX-015, is for retinitis pigmentosa, for those advanced patients. About 15% of the population, as I mentioned, given there are about 125,000 patients in the United States, that's about 20,000 patient population that we could treat. You'll note importantly that optogenetics has one other advantage in that its effects are likely to be beneficial independent of the underlying mutations. So where now in retinitis pigmentosa we recognize 60 or 70 individual genes and hundreds of different mutations, optogenetics should be applicable to all of those. The same preclinical program will support us moving into Stargardt's macular degeneration, which is a juvenile form of AMD, and we then hope to segue into the geographic atrophy market to treat those patients with established atrophy in time to come. So a few words about optogenetics. What is it, and how do we actually make this work in the retina? So it's a happy collision of a couple of disciplines. It's been known for about 100 years or so that single-cell microbes have the ability to move towards light. And they do this because they've evolved on their surfaces a channel rhodopsin, a channel protein, which is light-activated and results in a flux of ions. And in their case, they use flagella. It drives flagella transport towards the light. And then molecular biology has recognized these proteins, decoded them, and we now understand hundreds of them, and they've proved seminal in the neuroscience field, where you can express these channel rhodopsins in neurons and make them responsive to light, and therefore take control of those neurons, and you can map brain circuits, understand brain neurophysiology in that fashion. And more than 1,000 laboratories across the world are now using optogenetics. So a huge amount of work has been done to optimize these channel rhodopsins for use. Visual optogenetics simply says, well, there are surviving neurons in retinal degenerations of the eye, And so why not use these channelrhodopsins to make those light sensitive? And then incoming light from the environment would stimulate them, in this case the retinal ganglion cells in RP, and you'd get a formed visual signal. And that's been de-risked now. Multiple laboratories, multiple companies have taken this forward. And it looks like it works. It's an amazing technology, which you think is a little abstruse perhaps, but it does show promise. And the early phase, first generation optogenetic companies are seeing evidence of improved light sensitivity in their patients. There was a recent publication from one group in Nature Medicine recounting a patient whose vision has improved. So the technology appears to work. Ray Therapeutics have taken the best optogenetic protein that nature has produced, the best channelrhodopsin, further optimized that for characteristics that will suit patients' vision, and that's our RTX-015 approach. And here's a little bit of the data showing that. So mouse data here, not human data yet, but we have really very nice tools with which to measure the function, the visual function of mice, as you can see here. And so when we actually do this in vivo, we can see that compared with the first generation on the right-hand side, the black diamond, the first generation optogenetic proteins needed an awful lot of light. 1e16 photons per centimeter per second is about bright sunlight, whereas our crown protein, which is in the lead asset, RTX-015, is active right the way down to cone opsin ranges, the lower end, so street lighting at nighttime. And in this experiment, we're also able to compare competitors that are in the clinic head-to-head. And we can see that by comparison with all of the other competitors, our engineered protein is about 100 to 1,000-fold more light sensitive than anything else that has been assessed before. One important aspect of it is that most of these first-generation optogenetic proteins will need some sort of light intensification to get them to work, so-called goggles. And so all of those falling below the dotted line would need those. So Crown will be active in ambient lighting while retaining a very wide dynamic range of vision. This is our most important data set because with this particular protein's performance, we are for the first time seeing in our animal models the appearance, the creation in these blind animals of visual acuity and measurable visual acuity and contrast sensitivity. So in the treated mice in green here, you get a more than 50% return of visual acuity in these otherwise completely blind mice, and a near normal return of contrast sensitivity, which is hugely important for many of these diseases. We've compared this with our other head-to-head competitors, and we've not been able to demonstrate visual acuity responses in this. So really, it shows the potential performance of our optogenetic approach as we move towards clinic. We're also very pleased with our manufacturing as we move towards clinic. We've forged a relationship with Forge Biologics, who are moving straight forward with a commercial manufacturing process. So that will further de-risk our program and allow us to move very efficiently towards commercialization. Our approach is to deliver by intravitreal injection. We have a brisk and efficient rare disease development program, and we're moving into a registrational trial. We expect to see early proof of concept. That's one other thing here which is very interesting about optogenetics. In contrast to many of the other retinal degeneration clinical trials where you're looking at endpoints which look at the blunting, slowing of the rate of degeneration, which can take many years to detect. In these patients, because we're looking for a visual improvement, we should see that very soon, and that should be sustained as early as a couple of months. So when we look at our clinical trial design, we may well be able to declare proof of concept within about 6 to 9 months of beginning the study. The other beauty is that with what we're seeing with our particular approach is that we should see improvements in fully approvable endpoints, visual acuity, visual field, and mobility testing. So we're excited to hopefully move towards clinic in 2023 and hopefully bring realistic hope to patients with blindness of being able to see once again. Thank you very much for your time.